genes in that. So these are all the earlier projects what we have undertaken. So recently we are now at present we are working on marker assisted selection for DNV2. That is densovirus, that is flatchery, viral flatchery we call, so which infects silkworm. So here though compared to NPV this is not very high, but sometimes especially when during this season, so the flatchery is also more in the field conditions, especially in the field conditions. So already there are reports that they have added, it is a single gene is responsible for the susceptibility, resistant to the gene, that is non-susceptibility to DNV, NSD2 gene we call. So when this gene is not there or when this is mutated, the virus will not be able to enter into the silkworm, thereby the silkworm is resistant to this particular disease. Whereas there are some silkworms where this gene is intact, the whole gene is intact, so the silkworm is able to enter into the silkworm and thereby they multiply and cause the diseases. So in India there are also reports where they have screened the whole germplasm. We have our germplasm here, so we have screened. So out of so many germplasm what we are having, around 370 plus we have. So there some of them are completely resistant to DNV, whereas some of them are totally susceptible to DNV. So we have the list of all those things. So keeping that the earlier work whatever was done based on this, now we have for farmer for field oriented technology, what we are trying to do, we are trying to identify, we have already identified what are the silkworms are having this or not having this. So that we have taken, and then we are trying to develop new breeds which are resistant to DNV. So that work is going on especially in this lab, and she is my student Vanita. She is working on it, and she is a MSc student like you people. He is also doing his internship here. So we are working on that. We are working on especially bivoltane. We are not touching any of the multivoltane, though that is there in our profile, but we are not using it. So we are concentrating mainly on the double hybrids which are there in South India, because that is the ruling hybrid. So we have taken those parents and we want to develop a disease resistant variety, and we are concentrating on northeast, especially Berhampur, West Bengal, northeast region, where there are some bivoltane. There are some bivoltane silkworms like SK6, SK7 you might have heard. So those races also we are taking, and we are trying to make it resistant to diseases so that it survives against the DNV2 resistance. So this is all the work is going on here.
Thank you. Thank you so much, madam.
Thank you.
All of you.